Okay, up to this point, what we have seen is the metabolism of carbohydrates. And we've been focusing on skeletal muscle cells because the primary objective of skeletal muscle, of course, is to generate ATP required for contraction. Now, what we've seen up to this point is that skeletal muscles will perform a large amount of glycolytic reactions to process glucose into ATP. The glycolysis then, of course, will go into the TCA cycle, which will blend into the electron transport chain. Now, coupling these three sequences of events, we are able to oxidize glucose all the way down to carbon dioxide and generate a large amount of ATP in this reaction. Now, recall that the actin-myosin activity in skeletal muscle, once we have contractile events which causes those interactions, we are going to be hydrolyzing an enormous amount of ATP. The oxidation of glucose generates a large amount of ATP. However, that isn't always sufficient for muscle activity. So now what we're going to talk about are fatty acids. In this case, fatty acids will be taken up by skeletal muscle, and we need to transport this fatty acids into the mitochondria, because it is in the mitochondria that the fatty acids will be oxidized by a process called beta oxidation. Beta oxidation will generate an enormous concentration of acetyl-CoA, which of course will feed into the TCA cycle, followed by electron transport and generating a large amount of ATP. Now, the reason this reaction is so important is because if we just solely relied on the ATP generated by skeletal muscle from glucose oxidation, we may be limiting the amount of ATP available for contraction of muscle. By incorporating fatty acids, which contain a much greater percentage of energy stored as opposed to glucose, and compared to glucose, we can generate an enormous amount of ATP, much more so than we can with glucose. Now the question is how does the fatty acids get processed by skeletal muscle cells? Well, we will look in a later video that fatty acids are going to come from adipose tissue. Remember, adipose is our fat storage organ, and the adipose tissue will be subject to glucagon signaling. Now recall, glucagon is the hormone that is indicating to the body that we are in a fasting state. Once we are in a fasting state, recall that skeletal muscle has no access to glucose outside of its cell, but has to rely on the glucose that is coming from glycogen only. Our glycogen stores are limited in skeletal muscle, and so the fasting state releases fatty acids from adipose tissue, which now can supplement energy production in skeletal muscle along with the glucose in the limited stores. So we'll take a look at how glucagon signals in adipose tissue in a later video, but for now, suffice it to say that the adipose tissue will be releasing the fatty acids into the blood. Now, these structures of fatty acids, recall, are a long hydrocarbon chain, and they end with a carboxylic acid. So here's our standard structure of our fatty acid. The N is typically going to be of a long chain or very long chain. So this N will probably be roughly 14 to 16 carbons long. Now, this fatty acid in the blood is not traveling by itself because, remember, this is a hydrophobic molecule, so it's actually coupled in complex to the largest protein concentration in our blood, which is albumin. The albumin binds the fatty acid and transports it in the blood to the tissues that will process the fatty acid. Now, the first thing we need to do is get the fatty acid inside the skeletal muscle cell. So if this is our plasma membrane, recall that this structure of a fatty acid is going to not be able to simply diffuse through the membrane. Although the long chain hydrocarbon chain of the fatty acid is hydrophobic and completely miscible with the bilayer of the membrane, we have a powerfully charged group here which will prevent transport by simple diffusion. So therefore what we need is a transporter. And this transporter is called FATP, and that stands for fatty acid transport protein, FATP. Now this fatty acid transporter is going to be part of the mechanism which is called facilitated diffusion. And simply, facilitated diffusion refers to protein-mediated transport across a bilayer. So this is going to be working on a concentration gradient of high concentrations of fatty acid in the blood and lower concentrations in the cell. So this protein will allow the passage of fatty acids into the cell. Now, recall one of the properties of facilitated diffusion is the possibility of reverse transport. Once this fatty acid is transported into the cytoplasm of the skeletal muscle cell, it is possible for this fatty acid to be translocated back out into the blood. So the first step we need to do is a reaction where we are going to trap the fatty acid inside the cell. The enzyme that's going to perform this trapping reaction is called fatty acyl-CoA synthetase. This enzyme is going to hydrolyze ATP as the energy source to drive this reaction. And this reaction is so important, we are not going to be just generating ADP, but we are going to be hydrolyzing both of the high energy phosphate bonds on the adenosine energy molecule. So the first step is to hydrolyze ATP into AMP plus pyrophosphate, and then a pyrophosphatase enzyme will cleave this pyrophosphate unit into two inorganic phosphates. Recall these are high energy bonds, and so instead of releasing the energy of one high energy phosphate, we are releasing the energy of two high energy phosphates. That gives us a large negative free energy change to drive the reaction. Notice the name of the enzyme also has CoA in the enzyme. So in this reaction we are going to be adding a coenzyme A to the fatty acid. So the final structure we are going to be making is called a fatty acyl-CoA. This fatty acyl-CoA structure now is different than the fatty acid that was transported by the FATP protein. Therefore, this is not recognized by the protein, and so this will be trapped inside the cytoplasm, and the reverse reaction by the FATP is not possible. Now, another important thing to note is that this, because we are releasing a lot of energy from the hydrolysis of ATP, this is a high energy bond. It's a high energy thiol intermediate. So now once we have the fatty acyl-CoA intermediate here, we need to transport this to the mitochondria. Now recall again, we have a chemistry problem here because the cytoplasm is an aqueous environment and we have a rather hydrophobic molecule with this long hydrocarbon chain. So therefore we need an escort protein. And the escort protein is going to be shielding the hydrophobicity of the fatty acid during its transport to the mitochondria. And so the way it does this is it's going to envelop in the interior of the escort protein the hydrophobic domain. And the escort protein is called FABP, and this stands for fatty acid binding protein. The simple need of this protein, again, is to shield the hydrophobic domain of the fatty acid to allow transport through the aqueous cytoplasm to the mitochondria. Now let's look at the mitochondria. This is the outer mitochondrial membrane. And the fatty acid is going to be delivered to the outer mitochondrial membrane. And recall that the outer mitochondrial membrane has a certain level of porosity to allow medium-sized molecules through by simple diffusion. So what we have on the inside of the mitochondria is another protein that's embedded in the inner mitochondrial membrane. The active portion of this particular protein, of this enzyme, is facing the intermembrane space. So this will represent the inner mitochondrial membrane. And this, of course, between them is the intermembrane space. Now this particular protein is going to be performing a reaction that is going to facilitate the transport of the fatty acid into the interior and the matrix of the mitochondria. This particular protein is called CPT1. And CPT1 stands for carnitine, palmitoyl, transferase 1. Now the activity of this enzyme is going to take the fatty acyl-CoA structure and it's going to perform an enzymatic reaction that is going to substitute carnitine for the CoA group. So in the inner mitochondrial space, we are going to be releasing CoAsh, and we are going to be attaching to the fatty acid a small molecule called carnitine. Now recall, this is a high energy bond. This thiol bond is a high energy structure, and so this high energy structure will be captured between the carnitine and fatty acid. So we are essentially not losing any energy in this process. Now the next step of the reaction is that we have carnitine, fatty acyl carnitine, is going to be recognized by another protein that's embedded across the inner mitochondrial membrane. Let me properly balance our chemistry here. Showing carnitine entering the reaction, CoAsh leaving the reaction. And so the fatty acyl carnitine is going to be transported into the matrix of the mitochondria from the inner membrane space by this enzyme. This enzyme is called the carnitine acyl carnitine translocase. So this is a translocase enzyme that will recognize this fatty acyl carnitine and transports it into the inner mitochondrial matrix. Now once this is transported in here and we have our fatty acyl carnitine structure, we need to properly prepare this molecule for the next steps of fatty acid oxidation, which of course is beta oxidation. So in order to do that, we have another enzyme also embedded in the inner mitochondrial membrane. This enzyme is called CPT2. As you can imagine, it's the same type of enzyme. This is called carnitine, palmitoyl, transferase 2. And the job of this enzyme is to take the fatty acyl carnitine structure and processes it back to a fatty acyl CoA. In doing so, of course, we have to enter fatty acyl CoA, CoAsh, into our reaction, and what's coming out of the reaction is carnitine. Now, once CPT2 performs this reaction, restoring fatty acyl CoA structure in the matrix of the mitochondria, then this structure can go on to be oxidized by the reactions that we'll see in the next video called beta oxidation. Now, to complete this cycle of events in order to get fatty acid into the mitochondria, we have to make sure that we balance our chemistry. Notice that carnitine by CPT1 is entering the reaction and being delivered into the matrix of the mitochondria. If this was not processed so that we could remove the carnitine from the matrix back into the inner membrane space, we would have a carnitine depletion very rapidly. So in order to balance this chemistry out, what we're going to do is we're going to use the translocase enzyme, the carnitine acyl carnitine translocase, to translocate carnitine from the matrix into the intermembrane space so that these reactions by CPT1 can continue. So this translocase by this is an antiporter. It is a 1-to-1 antiporter, so it translocates fatty acyl carnitine in one direction and carnitine in the other, and that balances the carnitine out to prevent a carnitine deficiency.